I'd like to talk to you briefly in the next 20 minutes about a shared journey involving colleagues at the International Center for Diarrheal Disease Research in Bangladesh and our group here at Washington University, a journey that is predicated on hope, tempered by humility, given the complexity of the problem, but whose goal is to develop new therapeutic approaches for treating a vexing, pressing, and in many respects, tragic global health challenge, childhood undernutrition. And the journey that I'll describe to you is a multi-step journey that illustrates many of the steps that need to be undertaken when trying to understand the role of the microbiome in disease pathogenesis and developing new therapeutics. And that journey includes, as I will describe, defining normal to begin with, developing metrics for quantifying deviations from normal and determining whether those deviations correlate with disease incidence and severity, doing tests of causality to determine whether alterations in the microbiome are an effect or a cause of disease, identifying therapeutic targets, and once therapeutic targets are identified, developing therapeutic interventions that are affordable, that are culturally acceptable, that are scalable, and most importantly, effective and safe. So let's begin this multi-step journey together. As you all know, childhood undernutrition affects a large number of children worldwide, leading cause of death in those under 5. A lot of epidemiological studies showing it's not due to food insecurity alone. Current therapies have reduced mortality, but they've had more disappointing effects in overcoming the long-term sequelae of undernutrition, which include stunting, poor immune responses, including to vaccines, and neurodevelopmental delays. So we're certainly missing things about our knowledge of disease pathogenesis. I'll also add in this particular slide something that you all know, which is that the definition of undernutrition is largely based on anthropometry, with stunting defined as an LAZ score and impaired bondral growth by a weight for length or WLZ score.
Sorry to interrupt you, Dr. Gordon. We cannot see your slides. Could you
Oh,
share?
I'm so sorry. I'll try to share again.
They're not visible. Yes,
Can
they
you
are.
see it now?
But I think you need to do a slideshow. Yeah.
See it
Yes, there.
now?
Thank you.
Okay, thank you for telling me. So I'll just say again that the definition of undernutrition is based on anthropometry, with impaired linear growth being expressed as a LAZ score and impaired pondral growth or wasting by a WLZ score. But that's a fairly coarse definition of biological state or the biological states of undernutrition. As you'll see, it's going to be very important as we try to connect the microbiome to growth faltering to be able to have a deeper understanding of systems physiology. So our central hypothesis that we've been pursuing as a team is that there is a definable normal program of gut microbial community assembly and development that begins at birth, and that this program is disrupted in children with undernutrition, and such a disruption is not simply an effect, but a contributing cause to undernutrition. The corollary is that healthy growth of infants and children is linked to a program of co-development, not only of the gut microbiota, but also of host organ systems and subsystems. So to pursue this hypothesis, we'll go through this multi-step journey I alluded to, which is to initially define normal in a population where the burden of disease is great. In this particular case, I'll illustrate that with a birth cohort study that was conducted in an urban slum in Dhaka, Bangladesh, where children were enrolled at birth, and there was monthly fecal sampling of each child from that period of time all the way through initially 24 months, but extending to 60 months. At each monthly time interval, anthropometry was also obtained. And for those children who had consistently normal anthropometry, we focused on the fecal microbiota and trying to understand which bacterial taxa changed in their representation as a function of time. To do so, we did PCR of fecal DNA, amplifying a phylogenetic barcode of life, of bacterial life, the 16S ribosomal RNA gene. Sequencing these amplicons yielded a large data set of bacterial taxa represented in the developing microbiota of these children with consistently normal, healthy growth. It was a massive data set, and we had to apply machine learning algorithms and also statistical approaches to identify covariant networks of bacteria, to identify a small number of organisms that could describe this evolution of the microbiota in these healthy children. These taxa are referred to in this particular slide as an eco group. They describe normal community development in healthy members of this birth cohort. And it also told us that development of the microbial community in these healthy children is largely complete by the end of the second postnatal year. It also allowed us to look at children with undernutrition and, again, to develop this metric for quantifying deviations from normal. How were these normally appearing organisms represented in children with undernutrition? The answer was that the children with undernutrition had communities that looked younger based on the representation of these age discriminatory bacterial taxa than you would expect based on their chronological age. And this immaturity, if I can call it that, was not substantially repaired by currently used therapeutic foods. Of course, none of these foods were developed based on a consideration of the developmental biology of the gut microbiota. So in a sense, these children had a persistent developmental abnormality affecting one of their organs. In this particular case, that organ I'll refer to is a microbial organ, the microbiota. The next step in the journey, having defined a way of describing normal development and developing a tool to quantify deviations from normal. And by the way, the degree of immaturity of the microbial communities was worse in children who had more severe undernutrition than those who had more moderate forms of undernutrition. The third step is a test of causality. Is this immaturity or this perturbed development of the microbiota a cause or simply an effect? So in order to do this test, we turned to animals reared under entirely sterile condition in a so-called notabiotic isolator. These animals were colonized with intact, uncultured microbial communities from children who either had healthy growth phenotypes as defined by anthropometry, or children who had growth faltering, again defined by anthropometry. These children were all chronologically age matched, and for each donor fecal sample, a group of animals were colonized with the constituent organisms. All animals from all groups were fed the same diet, a diet representative of the population that we were studying. And what was found in this test of causality is that microbial communities from children with growth faltering transmitted to recipient notabiotic animals many of the features of undernutrition, which is impaired weight gain, perturbations in bone biology, perturbations in metabolism in a number of tissues, and altered immune function. So you could then take this group of animals, which represented the population that we ultimately wanted to treat, and survey all the microbial communities installed in all the animals and use machine learning algorithms to identify organisms whose representation correlated with different aspects of growth, i.e., to identify growth discriminatory strains. That was done. These organisms, it turns out, were not appropriately represented in the developing communities of children with growth faltering, and they became our therapeutic targets. So representative, we hoped, preclinical model as a platform for identifying therapeutic targets. But the therapeutic approaches that we had to use had to be culturally acceptable, affordable, scalable, and that wasn't going to be the case if we were to take these growth discriminatory organisms and culture them as we did, sequence their genomes, and create a next generation probiotic consortium. It would have been much too difficult from a technical standpoint to manufacture such a consortium. The cost would have been prohibitive, and also the regulatory barriers to be able to gain approval for that consortium, at least at the time when we were beginning this journey, were too formidable. So we turned instead to food, particularly the food consumed by these children where they lived. These complementary foods, we hypothesized, contain key nutrients, or at least some of these foods might contain key nutrients that could be utilized by these growth discriminatory, growth promoting organisms in the developing gut microbiota of children with undernutrition. So our approach was to screen in these preclinical models, i.e. animals colonized with the microbial communities of the very population that we wanted to treat, screen different complementary foods, initially singly to groups of animals to determine whether they could promote the fitness, i.e. increase the representation of underrepresented organisms, and alter the expressed beneficial functions of these targeted organisms. Such a screen was conducted initially with single ingredients, and then when we had hits, we combined them to determine whether multiple ingredients together would have greater effects on repairing the microbial communities from these children. And we envisioned that we would term these formulations microbiota-directed complementary foods, MDCFs. So such a screen was conducted. We had hits, and several of the MDCF prototypes, which we identified initially in gnotobiotic mouse models, but then subsequently in gnotobiotic piglet models that contain the organisms from the very population of children that we wanted to treat. These leads emerged and were subjected to an initial pre-proof of concept study involving children living in Mirpur, that urban slum, who had moderate acute malnutrition, MAM, a WLZ score between -2 and -3. These children were 12 to 18 months of age. It was a short study, 4 weeks of an intervention with one or the other of our MDCF prototypes, and another arm received a standard of care, a ready-to-use supplementary food. There was a run-in period where we obtained fecal samples and anthropometry. At the end of the run-in period, a blood draw was obtained because we wanted to develop a much more comprehensive definition of the biological state or states of these children prior to initiation of treatment. During treatment, which was done under supervision twice daily, we obtained a fecal sample, and at the end of the treatment period, again, a blood draw, a fecal sample, and anthropometry, and a 2-week post-intervention follow-up. We didn't expect, because of the size of the study, that is to say, the power of the study, only 15 children per arm, that we would obtain a statistically significant change in anthropometric measures of growth. But we did hope we would see which MDCF would be most effective in repairing the microbial communities, and perhaps which ones would also change the plasma proteome. In this particular iteration of such a pre-POC study, we used aptamer-based proteomics, that is to say, the use of DNA aptamers that could recognize each aptamer a specific plasma protein. In particular, 1,305 proteins that represented biomarkers and mediators of many aspects of host physiology. A lead MDCF emerged from such a study, and this is a nutritionalist view of what such a formulation looks like. The 4 complementary food ingredients in this MDCF, which we'll call MDCF2, were chickpea flour, peanut flour, soybean flour, and green banana. Lipids were represented as soybean oils, and there were vitamins and minerals in the form of a micronutrient premix. The standard of care, a ready-to-use supplementary food, contains none of these complementary foods, but rather rice, lentil, powdered skim milk, soybean oil again. And I want you to look at the bottom of this particular screen to see that the caloric density of MDCF2 is 15% lower than that of RUSF. So a larger powered, longer study was performed. Again, children 12 to 18 months of age with MAM living in Mirpur, twice daily administration of either RUSF or one of the MDCFs, specifically MDCF2. Over this 3-month period of time, fecal samples, anthropometry, and plasma was obtained. And at the end of the period of time, we looked for a change in the primary outcome measure, which was WLZ. It was very important to us, since we didn't know the full effects of microbiome repair, to extend the study of these children beyond the period of intervention. And we obtained consent to follow these children for 2 years with 6-month intervals between sampling of their fecal microbiota and their plasma proteome and the measurement of height and weight. And that was going to be very important, as you'll see in a second. The secondary outcome measures from this trial, and there were 60 per arm, 60 children per arm, was gut microbiome repair, the plasma proteome, in particular changes in the mediators of different aspects of growth. So this is the result. The calorically less dense formulation, MDCF2, created a more rapid increase in the measure of ponderal growth, WLZ. It also had a profound effect on the plasma proteome. In this particular case, we were able to measure 5,000 different plasma proteins in 100 microliters of plasma using this aptamer-based proteomic platform. And for each plasma protein, for all children, that is to say, children in both arms of the study, we correlated changes in the level of that protein with changes in WLZ. And in this way, we're able to identify 70 proteins whose abundances or levels in plasma positively correlated with WLZ. And they include plasma proteins that are involved in the regulation of musculoskeletal and CNS development, as well as metabolic regulation. There were also a series of proteins that were identified in this fashion, which were negatively correlated with WLZ, and those principally were biomarkers and mediators of inflammation. MDCF2, the microbiota-directed complementary food, produced a significantly greater increase in the levels of these positively correlated proteins and a significantly greater decrease in the level of these negatively correlated proteins compared to RUSF. The intriguing finding was that although there were no statistically significant differences between the two treatment groups in WLZ at the end of the 3-month intervention, during the next 2 years, we saw a significant improvement in linear growth as measured by LAZ. And that is an important lesson. We saw the plasma biomarkers and mediators of musculoskeletal development changing significantly during the 3-month intervention, but the clinical outcome measure became manifest only beyond that period of intervention. Now, what are the bioactive components of MDCF2? And how should we look at MDCF2 from the perspective of the microbes? Because they don't see labels like chickpea or peanut or banana or others. Rather, they see biomolecules. So together with Carlita Labrella, we looked at the polysaccharides that are represented in the complementary foods that were used to formulate MDCF2, as well as the foods that were used as ingredients with RUSF, and identified the principal polysaccharide structures that distinguish MDCF2 from RUSF. And I won't have an opportunity, because of limited time, to go into any discussion of this aspect of this project, which is critical, the glycobiology. But I will tell you how that information was critical in interpreting how MDCF2 worked. The other thing we did in looking at structure-activity relationships is turn from structure-activity relationships and interest in discerning them in the food formulation administered to the microbiome itself. So shotgun sequencing was performed of the fecal samples obtained from each child in each arm of the study. We had on average 8 fecal samples, all of which were frozen within 15 minutes of their donation by the child at liquid nitrogen temperature and maintained at minus 80 degrees thereafter in order to preserve not only DNA, but RNA and metabolites. And the shotgun sequencing, the DNA-based analysis, yielded metagenome assembled genomes. So the algorithms for assembling shotgun sequencing reads into the genomes of bacterial strains that are represented in the microbiome has advanced rapidly. And we were able to identify 1,000 of these bacterial mags in this particular population. The way we do that for each child, an ensemble of mags is identified. For all the children with all of their mag assemblies, we dereplicate to get these 1,000 mags that are shared across the population, and then look for the genes represented in these assembled genomes, annotate these genes to determine or predict their functions, metabolic reconstructions. We use an annotation platform that was developed by Andre Osterman and his colleagues called Microbial Community Seed, and then looked for those mags that were positively correlated with WLZ, that is to say, whose abundances positively correlate with WLZ. From the 1,000, we identified 75, and then asked which metabolic functions were enriched in the genomes of these positively correlated mags, and they were principally pathways involved in the utilization of various carbohydrates. And then we turn from DNA level analysis to gene expression analysis to determine which metabolic pathways in these positively correlated mags were induced or regulated by MDCF compared to RUSF. And the results were remarkable. This is a very complicated slide, and I want you to look to the right first in the bullet points. We're able to correlate WLZ responses with expression of genes in these carbohydrate utilization pathways whose representation were enriched in the genomes of positively correlated mags. And in particular, the carbohydrate utilization pathways that were involved in the metabolism of the polysaccharides we identified from analyzing MDCF2 were induced by MDCF2. So those were ways in which we identified the bioactive components of MDCF2. But there was even a more sublime way of doing this. It turns out that most of the differential expression of carbohydrate utilization pathways in MDCF2 treated children were linked to 2 mags, both of which were members of Prevotella capri. And there were 11 different P. capri mags in these collections of mags that we were able to identify, only 2 of which were positively correlated with WLZ. And these were the ones that had differentially expressed carbohydrate utilization pathways. These pathways could be seen from the perspective of polysaccharide utilization loci. These are clusters of genes distributed throughout their genomes, and each cluster contains a series of genes encoding enzymes that break down particular carbohydrate structures. And the conservation of these polysaccharide utilization loci is depicted in these slides in the black box as green colored polysaccharide utilization loci. And these particular P. capri mags had much more conserved pools than any of the other P. capri mags and served as a guideline so that we could culture from the well-preserved fecal samples the organisms that are represented by these mags. So we were able to culture P. capri strains that capture the genomic features of these transcriptionally responsive to MDCF2 mags. We then said to ourselves, okay, we have an idea of what the bioactive components of MDCF2 are. We have a better idea of the therapeutic targets of MDCF2. Could we gain better information, more direct information about whether these particular strains of P. capri were key drivers of growth response? And for that, we did reverse translation experiments. We returned to those notobatic mice that began our journey And we did the following experiment. We introduced defined consortia of genome sequence, age, and WLZ-associated bacteria that had been cultured from the very population where we had done the clinical trial. And we introduced those consortia of genome sequence bacterial strains into germ-free female mice that just had delivered their pups. So the recipient female mice would transmit those organisms to their pups. And we did so in waves of colonization, taking as the first consortium early colonizers of the gut microbiota, a second consortium representing later colonizers, a third consortium representing still later colonizers. So in the pups, we're able to reenact the normal pattern of assembly of the microbiota. The pups, in turn, received a sequence of diets that resemble the sequence received by participants in the clinical trial, ending with weaning onto MDCF2. There was an important difference between two groups of mice that were the subjects of this experiment. One group got the sequence of bacterial strains that included P. copri, and the other did not. So two groups of mice, those whose consortia did or did not include the cultured P. copri isolate representing the two mags, and the key findings are shown on the right. Mice that lacked P. copri had more impaired growth. That is to say, mice that had P. copri had significantly greater weight gain. They were more adept at processing MDCF glycans, and there were profound effects when P. copri was present on energy metabolism in gut epithelial cells. Interestingly enough, the spatial patterns of change in epithelial gene expression and metabolism were very distinctive, confined largely to enterocytes positioned at the base and middle regions of villi. Now, how generalizable are these effects? We're almost done with the presentation. I'll tell you that there will be soon published a paper that dealt with a similar trial design, except for in this particular case, the Bangladeshi children not only resided in Dhaka, but at a rural site, Kurigram. These children presented initially with severe acute malnutrition, underwent standard nutritional rehabilitation, but as is typical of the situation with children with SAM, after standard therapy, they emerged with moderate acute malnutrition. So these children with post-SAM MAM were then divided into two treatment arms, one with MDCF2 and the other with RUSF. The design of the experiment was identical to that I described earlier with primary MAM, and the outcome was identical. That is to say, those children who had post-SAM MAM treated with MDCF2 had superior ponderal growth responses, and in follow-up, superior linear growth responses. And as before, the mediators that we were able to identify in the plasma proteome of musculoskeletal development, CNS development, regulators of metabolic function, regulators of immune function, changed in a way that was significantly improved in those who received MDCF2 compared to RUSF. So in my final two slides, let me just summarize and look ahead. So the key challenge is to determine now the durability of the effects of MDCF2, long-term safety, its generalizability. So we have to test in other populations as we are. There is a study being sponsored by the Gates Foundation, WHO, and UNICEF that involves multiple sites, Pakistan, India, Bangladesh, as well as sites in Africa, Tanzania, Mali, and Niger, of children with MAM, but over a broader age range, 6 to 24 months. The tests we've done here are largely children in the second postnatal year. So is this treatment going to generalize over that age range? Remember the central hypothesis, it's co-development of the microbiome and hosts being linked to healthy growth. Another important challenge is to determine the malleability of the system. What's the biological state at the time of clinical presentation? So does the duration and severity of disrupted community development affect response to treatment? Is the chronological age when the developmental disruption of microbiota assembly first occurs going to influence outcomes? And finally, how about the clinical responses? It's going to be very important to judge response not only during treatment, but in longer term follow-up. So these studies have to be designed where each child represents her or his own control, but there has to be a level of community engagement and commitment on all parties so that this long-term follow-up is possible. We also have a unique opportunity to connect the dots because as we repair a microbial community, we have an opportunity to be able to determine how the representation and express functions of different components of a microbial community are linked to the operations of different host systems and subsystems. So returning an investment in mechanistic studies could yield critically biomarkers for stratification of disease population, better ways of evaluating efficacy, including durability, and also ways of predicting responses. Ultimately, better definitions of wellness for infants and children with point-of-care diagnostics, and also, we hope, more informed policies about complementary feeding practices, policies that are informed by knowledge of the developmental biology of this microbial organ. Thank you so much for your attention, and it's a privilege and a pleasure to be able to talk to you today.